Good morning, everyone. Thanks for showing up today for our webinar on the CI340, shedding light on photosynthesis, how researchers are using photosynthesis in 2020. Our webinar will be focused on the CI340 handheld photosynthesis system. We will be having a guest speaker, and we will also be going over some research done in 2019 and 2020. I want to introduce our webinar moderator for today, Susie Truitt. She is our distributor manager. Many of you may have already had contact with her. She has been with CID Bioscience for 8 years, going on 9 years. And she will be posting links to all research articles and any other important links that you may find useful in the chat box. My name is Eric Munoz. I'm an application scientist here at CID Bioscience and Felix Instruments. I have a Bachelor's of Science in Biohealth Sciences from Oregon State University. Prior to working for CID Bioscience, I worked as a chemist and DNA analyst in the dietary supplement industry. For the agenda today, we'll be doing an overview of the company, as well as the CI340 instrument. We'll be going over the features and specifications. Then we will have our guest speaker, Dr. Stephanie Wedryk. She is the Director of R&D at Arcscreen Technologies. She will be presenting on some of the research that she conducts over at Arcscreen Technologies and how she utilizes the CI340 in her research. Then I will be speaking about some of the current research that we are doing, or that researchers have been doing using our instrument, and a little bit of detail. And then afterwards, we will be concluding with a Q&A session. So a little company overview. We were founded in 1989. We applied more than 30 years of experience in plant research instrumentation to serve the research community and commercial needs. We pride ourselves on creating non-destructive measurement tools to help researchers acquire consistent and high-quality data. Our instruments are known for their ability to produce instant, accurate results. Their portability, durability, and data transparency are many of the benefits that our instruments provide. All of our product line is engineered, tested, and manufactured under the same roof in our headquarters in Cannabis, Washington, United States. A quick overview of the CI340. It's an essential utility in the lab and field. CID Bioscience provides the CI340 handheld photosynthesis system. It's a light, handheld, and not requiring backpack of gear. The instrument is field ready, weighing less than 3 pounds. The CI340 takes non-destructive open or closed system measurements, utilizing a variety of sensors, including infrared carbon dioxide analyzers, or IRGA, a humidity analyzer, also uses a PAR, or photosynthetic active radiation sensory data, or PAR, and multiple temperature sensors to measure leaf, air, and internal component temperatures. All of this data is able to give you information such as photosynthetic rate, which is determined by measuring CO2 before and after it enters the leaf chamber to calculate the rate of CO2 assimilation by a known leaf area. It also provides you the transpiration rate, which is determined by measuring water vapor before and after it enters the leaf chamber to calculate the rate of water vapor flux per one-sided leaf area. It also provides you stomatoconductance measurements, which are calculated by measuring transpiration rate as a function of leaf temperature. The CI340 is easily operable as either an open and closed system. In the open system measurement, the incoming gas is drawn from the ambient or control module altered air and exhaust returns to the external atmosphere. During the closed system measurements, the air is recirculated from the exhaust back into the chamber, closing the loop. The CI340 is designed to measure both absolute and differential readings. In absolute mode, the CI340 measures gas concentrations from a single source. For differential measurements, which is the most common, the concentration of gases from both the inlet and the exhaust are measured. This will give you the net photosynthesis rate. Due to the large variety of research, we have 11 chambers for whatever you may be studying, as well as 4 additional modules that give some optional flexibility in your studies. Gas exchange measurements provide direct measure of the net rate of photosynthetic carbon assimilation, carbon dioxide exchange systems use enclosure methods, where the leaf is enclosed in a transparent chamber. The rate of carbon dioxide fixed by the leaf enclosed is determined by measuring the change in the carbon dioxide concentration of the air flowing through the chamber, as I stated before. But just to reiterate, we measure carbon dioxide assimilation in a single leaf in order to investigate whole plant characteristics. I've outlined some of the CI340 features. As I said, it's very portable, just weighing in at under 3 pounds. It's non-destructive. It can be used in open and closed system measurements. An open system, you would be utilizing both the inlet and exhaust measurements to calculate the differential. And a closed system measurement, you're closing the loop, so all the exhaust is being recirculated back into the system. The CI340 utilizes an ERGA analyzer to measure CO2. It also measures H2O and humidity, and utilizes PAR sensors as well. As I said before, we have 11 interchangeable chambers and 4 additional modules that can better control system environments. The calculated data is shown in real time on the display and saved in data. It saves to an external mini USB, I mean. The calculated data includes net photosynthesis, transpiration, and stomatoconductance. If you can download the manual by visiting our website, and it's important to mention that you can actually download translated versions of our manual from our website. 20 languages are supported. In order to do so, just visit our website, select the instrument that you are interested in. There will be a documents, a resources tab where you can select the manual. Once you open up the manual, it'll open up a Word document, and in the Word document, you can select the language that you want the manual to be translated into. Some of the CI340 chambers, these are the standard chambers. The first one is a 25 by 25 millimeter chamber. The second is a 55 by 20, and the third is our largest standard chamber, 65 by 10 millimeter. These are all used in open system measurements of trees, shrubs, and herbs with small and broad leaves. Here are a couple of our succulent and conifer chambers. They're the smallest one, the smaller of the two is 25 by 90 millimeters, and the larger of the two is 50 by 70 millimeters. These are also used in open system measurements of succulents and small needle conifers. The flaps, the orange flaps here, you can see allow needles to be placed non-destructively in the chamber while also sealing the chamber. Here are a few of our closed system measurement chambers. So these are used for measuring leaf or fully contained materials. Internal fans run when the CI340 is connected, allowing for measurements to be taken of a homogenized atmosphere, regardless of the increase in volume. So the containers are suitable for respiration studies or for a large variety of species. The smaller of the two, excuse me, I'll go back. The smaller of the two is a quarter-liter leaf chamber. As I said before, it's used for closed system measurements of long, narrow leaves. And the bigger of the two is a half-liter leaf chamber. Again, these are both used for closed system measurements. Lastly, here are a few of our other chambers. These are used for measuring cacti, also soil, and this one is a custom-made one. For the cactus leaf chamber, the window size is 14 by 14.6 centimeters squared. It measures the leaves of cacti. This one here is used for soil respiration, which include measurements of CO2 flux from soil. And the last one in the bottom right corner is the canopy chamber attachment interface between the chamber and instrument, which allows users to design and build their own custom chamber. Here's the image of our four optional modules. The four modules include a light intensity module, CO2 and H2O concentration module, temperature module, and a chlorophyll fluorescence module. The environment in the leaf chambers can be controlled using these separate control modules. The light module allows researchers to adjust the light intensity received by the leaf in the chamber. The CO2 and H2O concentration module allows researchers to set or adjust the CO2 and H2O concentrations in the chamber in order to investigate related physiological changes. The temperature modules allows researchers to adjust temperature in the chamber to evaluate fluctuations in the rate of photosynthesis relative to high or low temperatures. And lastly, the chlorophyll fluorescence module measures fluorescence simultaneously with photosynthesis to provide researchers with the information about changes in photosynthesis efficiency and heat dissipation from a leaf. This brings us to the applications using the CI340. These are some of the 4 main applications, beginning with phenotyping and genotyping, where you can track changes in phenological phases as well as genotype studies. The main and most common one application using our CI340 would probably be characterizing stress responses. You can track changes using gas exchange caused by environmental stress. Some of the studies that I'll be talking about will mainly focus on this application, characterizing stress responses. The next is maturity and aging. It's commonly used to track plant maturity, aging, and dieback, and how photosynthesis rates varies within these 3 stages. And lastly, the CI340 is a really powerful and great educational tool that is very helpful and useful in many plant science labs. But beyond these 4, there are also many non-traditional uses for a handheld photosynthesis system. Other non-photosynthesis uses include anything that releases or absorbs carbon dioxide or water, such as soil and insect respiration. Carbon credits and climate change can be studied as well by measuring the photosynthesis, carbon dioxide, and water exchange. I'd like to introduce to you our first guest speaker, a little technical difficulties. As I was saying, we have our guest speaker today, Dr. Stephanie Wedryk. I would like to introduce her to you. She is the Director of R&D for Arcscreen Technologies. She graduated from Ohio State University with a PhD in horticulture and crop science in 2011. Her PhD research focused on organic weed management and urban vegetable production. She worked for a large agricultural cooperative focused on product development for several years. She joined the Arcscreen Technologies team as Director of Research and Development in October of 2017. As you will probably hear, she's super excited about developing innovative products for cannabis growers. And without further ado, she will be presenting the next few slides.
Thank you, Eric, for the introduction. So, as you mentioned, I am the Director of Research and Development at Arcscreen Technologies, and just want to give you a really brief overview of the company, and then we will get into how we use the CI340 and how it's really helped us in our research and product development efforts. So Arcscreen Technologies, we provide professional cultivation solutions for cannabis growers, and our core focus are indoor and greenhouse cannabis growers in the United States and Canada. Most of our customers are working in the high THC versions of cannabis as opposed to hemp varieties which have low THC. We provide nutrients, pest management products, substrates, and various other additives to cannabis growers to help them through all stages of their cultivation cycles. And the key piece of what we do and what the research and development team does is our product testing platform. And all of our products go through rigorous testing and screening to ensure that they will work for the growers that utilize them. And in order to execute this product testing platform, the CI340 has been absolutely critical. And it has really helped us to understand the products and understand the plant responses to them. Next slide. So this is just a really brief overview of our product testing platform. And I've highlighted where we use the CI340. So we're really using it in our two main phases of testing on plants. So when we ideate a new product concept, we test it on plants immediately, and the CI340 we utilize in those trials in order to determine what type of response is this product even giving us. We use it really kind of as a screening mechanism at that phase to see, does it have any appreciable effects on photosynthesis, transpiration, or stomatal conductance? And that can give us some clues as to what type of product we're dealing with and what type of responses people might be able to expect. And then when we kind of get to phase 3, this is when we're actually testing a commercially viable product, and we really want to understand the different rates the product might be useful at. Does it work across multiple cultivars? So in cannabis, the breeding has not been standardized as it has been in other crops. And so the world of cultivars is very, very diverse. And so we really want to make sure that there are consistent responses, and we utilize the CI340 to be able to as one of our screening mechanisms in that regard. And it also helps us to kind of monitor plant health. We run lots and lots of trials, and we'll kind of get into how we grow here in a second, but we run lots and lots of different trials. And, the CI340 also helps us to kind of monitor the differences between those different trials. And we utilize a lot of different plants in our testing process. And the CI340 has really been instrumental in helping us to guide our ability to select the best performing products. So I don't have a slide necessarily on this, but I'll take you through the narrative that so give you a little background on how we grow at our R&D facility. So we operate in a facility in the state of Colorado. It is a fully licensed commercial growing facility. So we have a lot of advantages in that we are able to really test these plants as how growers see them. And this is a completely indoor grow. Most of our experiments are conducted under metal halide or HPS lights, and cannabis has two very distinct growth cycles. There's a vegetative cycle, and in our facility, the vegetative cycle is grown under metal halide lights. And then it moves into a flowering cycle, and in that flowering cycle, we're growing under our high pressure sodium lights. And the proliferation of flowers in cannabis is photoperiod sensitive. So when it enters that flowering cycle, there's 12 hours of light, 12 hours of dark. And we do pump in additional carbon dioxide into the facility. And this is all hydroponically grown. We grow in a cocoa-based substrate. So those are kind of the growing conditions under which we're working. So we do find, a lot of dependency on those growing conditions. And so just to highlight a little bit of some of our data, in the top left-hand corner, we tested a product that we were hoping would give an improved yield response. And we used the CI340 to understand the impacts on photosynthesis and determine whether was some of that yield enhancement driven by improvements in photosynthesis. And this was just done during the flowering cycle. And in the very beginning, the untreated and the plants treated with the product were nearly identical in their net photosynthetic rate. And over time, you can see that generally the trend is that there was a higher photosynthetic rate due to that product than with the untreated. We do have a little blip here during week 6. And you know, that happens in research, right? We see those types of anomalies. And one approach we've taken to try to mitigate those is we understand that by taking multiple samples within, you know, a rep or a treatment can really help to tighten up some of this data. And one nice thing, the CI340 has a screen that allows you in real time to see what your measurement is. And just when we're taking those measurements, it's a really nice feature so that we can then determine, you know what, that doesn't seem quite right. Let's take another one, and let's take another one and see if those are repeatable results. And it allows you to do that quite easily, which is really, really nice. The other thing I mentioned is that the CI340 has really been helped us to understand overall plant health and understanding trial consistency. So we repeat our trials multiple times. And because we're in a commercial growing facility, every cycle of plants is always a little bit different. And, you know, that helps us to understand kind of the strengths and weaknesses of what we're doing. But we also want trial consistency, that if things are too variable, we aren't really going to understand our products, and we aren't really going to understand what's happening with the plant. So it's been a great tool for us to be able to understand how different trials perform, as we've kind of illustrated in the graph on the lower right-hand side of, you know, you can see there's kind of a similar trend over time, but that there was some inconsistencies between those two trials. And you know, as Eric mentioned, some of the main uses of the CI340 phenotype, I don't have it illustrated here in the data, but we do see some pretty large differences between the different cultivars grown under the exact same conditions at the exact same time. Stress response has been a really big one for us. That's where a lot of our products have been focused, is on stress response and how they can mitigate stress for enhanced yield aging. So we take these measurements every week throughout the flowering cycle, which tends to be about 8 weeks. And so you can kind of see the overall trend in how photosynthesis changes in cannabis. Part of it is that the plant gains quite a bit of growth, especially during the first 3 to 4 weeks of the flowering cycle. So it gets closer to the lights. And so, you know, that will change the photosynthesis rate as well. And, you know, we've been really pleased with it in helping us monitor product performance, being able to tie it to any yield improvements. And, you know, sometimes we don't see yield improvements. But this, by monitoring photosynthesis, we've been able to see if, you know, there's something there, but because yield is such a complex trait, you know, the photosynthesis changes can give us a clue as to whether we are actually having an impact. So that's kind of a general overview of how we're using it, really diving into products for cannabis growers. Our focus has not been so much on cannabis biology in and of itself, but I guess I'm willing to take any questions if people have any or comments.
Yeah, so at the end of the presentation, we'll have a Q&A session. If you have time, Stephanie, I would encourage you to, I'm sure some of the researchers in attendance would probably have some questions for you. I did see one question. If you wanted, you could answer that now. Or if you want, you can also wait till the end. Any preference?
Is it the, I see a couple different questions. I'm willing to answer them now if people are interested.
Okay, yeah, we can, since your presentation is fresh in our memories, I think this one here, in Flower Week, your result is in zigzag. Is time important for this measurement?
So time is, I guess, clarity, time of day, is that kind of what the person is getting at?
I'm a little unsure.
If time of day is what they're referring to, yes, it is incredibly important. And we have learned that over time. We standardize. So because we're inside, lights come on at different times. We do allow for, after lights have come on, we like them to be on for a minimum of a couple of hours before we ever take a measurement. Because obviously when lights first come on, photosynthesis will be very different than after they've been on for several hours. So we have standardized that.
Excellent. Sanjay is also asking, in the above graph, there's no standard deviation. So how much sample requirement is for good results?
So yeah, so no standard deviation. We do have that information, they're just not in these graphs. And what we do is pretty much every treatment is getting, we have usually 4 to 5 replications per treatment. And then these are also averaged across 2 different cultivars. And these are really just for illustrative purposes. You know, in our product development process, we break the data down more than this. And then we also tend to take more than 1 sample per plant. So we'll take a sample on a plant, get the number, and then we'll just take it again right afterwards to try to ensure that consistency. But I mean, dealing with plants is always dealing with a lot of variation.
Yeah, that's a great answer. An anonymous attendee is asking, have you found any relation between photosynthesis rate and yield in cannabis? Does it apply with all varieties?
We have found some correlations with increased photosynthesis and increased yield. And we have found that across multiple different cultivars as well. But, you know, it's not always consistent. Yield is a complex trait. So sometimes increases in photosynthesis don't always translate to yield, or a yield increase is, you know, due to some other factor.
Great answer. Arthur is asking, do you use chlorophyll fluorescence to judge your products? If so, which parameters do you use?
So we do have a separate chlorophyll meter that we use to measure, and we are looking. It's from a different company, and they have their own algorithm for what their numbers mean, and they correlate it to both SPAD and total chlorophyll. So we look at that company's their proprietary number as well as the correlations to SPAD and total chlorophyll.
Great. I hope that answered your question, Arthur. Heather is asking, not really directly about the work, but have you tried using LED lights to reduce energy inputs? So
there is one room in this growing facility. So we have separate rooms in the growing facility. 2 of the flowering rooms are under high pressure sodium. 1 of them is under LED lights. And we have done a little bit of work in the LED room, just kind of some observational stuff, not really, you know, replicated randomized trial designs. And we just very observationally, we have found a little bit higher photosynthetic rates in those rooms. But, you know, again, we are operating in a commercial growing facility. So we have to kind of work with what the facility offers us.
Perfect. We're just going to take one more question. I know Stephanie is really busy, and we also have to continue with the presentation. So the question was, is your experiments in controlled conditions, or are they in ambient conditions?
So they're ambient to what the happens in that commercial facility. Since we are completely indoors, this is not a greenhouse. I mean, it's completely indoors. You know, the environments are much more highly controlled than you would find if you were doing these measurements outside, but we are not in a growth chamber setting or something like that.
Great answer. Thanks, Stephanie. I appreciate that, answering those questions on the spot. Typically, we do these questions at the end of the presentation, but I think we'll go ahead and just break the mold for this one time.
I will be sticking around if there's more questions towards the end too.
Okay, excellent, perfect. Appreciate that. So if anybody has any extra questions, I want to reiterate to use the Q&A chat box and not use the actual chat box for questions. If you have more questions for Stephanie, you can go. She will stick around at the end to answer any more questions that you may have. So continuing with the presentation, I have a few case studies that I want to present from research using our CI340. The first of which is transcriptomic response is more sensitive to water deficit in shoots than roots of Vitis riparia. So a little excerpt from the abstract is a little background information. Drought is an important constraint on grapevine sustainability. Vitis riparia is widely used in rootstock and scion breeding, and it has been studied in isolated leaf drying response studies. However, it is essential to identify key root and shoot water deficit signaling traits in intact plants. This information that they received or that they got or studied will hopefully aid improved scion and rootstock selection and management practices in grapevines. So the method that they use for the CI340, they use the calculated net photosynthesis rate and stabenoconductance. It was measured with the CI340, and the instrument was used just prior to measuring stem water potential. Gas exchange measurements were taken using leaves on separate shoots, but at the same node position as the leaves used for water potential. The photosynthetic measurements were made in one grapevine from each replication at the same time point, which was 7 days and 14 days. The leaf chamber that they used was the 11 centimeters squared of the total leaf area, and the meter was operated under open conditions. The leaf and air temperature were also measured, and they also had an optional module, the CI301 LA light attachment, which was used to photosynthetically to maintain the PAR at over 2,400 micromoles. The results that they obtained, they had strong correlation, an R of 0.9 between stem water potential and photosynthesis. So the higher the water potential, the larger the rate of photosynthesis. Shoot elongation, photosynthetic rate and somatoconductance were significantly reduced in water deficit treated shoots and roots in comparison to the well-watered grapevines. The RNA sequence analysis indicated greater transcriptional differences in shoots than in roots. I believe that this is in contrast to other studies where there's higher transcriptional differences in the roots than in shoots. But in this case, shoots had higher transcriptional differences with 6,925 compared to 1,395 genes differently expressed, respectively. The larger number of differentially expressed genes found in water deficit shoots indicated that the shoot had a greater response to water deficit than the grapevine root. Susie will be sharing the link to this study in the chat box below. Again, I want to reiterate to please keep the chat box clear of any questions so that all these links are easily manageable and you can follow along with the presentation. The next study is alleviation of salinity stress by hydrogen peroxide and nitric oxide in tomato plants. This study was done from the Department of Horticulture Science, College of Agriculture and Agricultural Sciences, and Natural Resources from the University of Khuzestan in Iran. So a little background, salinity is one of the major abiotic stress factors limiting plant growth and productivity, particularly in arid and semi-arid climates. So hydrogen peroxide and nitric oxide are important signaling molecules in the plant response to abiotic stress. The research, in this research, the effects of foliar sprays using hydrogen peroxide and sodium nitroprusside as the nitric oxide donor. They researched the effects of the foliar sprays on alleviation of varying salinity stresses, different concentrations of sodium chloride, which was investigated using tomato plants. The results showed the photosynthetic attributes, plant water relations, and membrane stability index were decreased by the sodium chloride treatments. Using the exogenous hydrogen peroxide and nitric oxide application, this enhanced the salt stress tolerance in tomato plants by improving the photosynthetic efficiency. The results were positively reflected by the increase in plant growth under salinity stress conditions. The results of the study described that under the adverse conditions of salinity stress, that hydrogen peroxide and nitric oxide could activate the photosynthetic system and improve the physiological attributes in plant growth. I don't know if you can see in the images in the PowerPoint, some of these graphs show total chlorophyll. So you can see that as the salt concentration increased, that total chlorophyll decreased, but the control unit, of course, well, all treated plants and the control decreased as the concentration increased. However, you can see that the treated plants with hydrogen peroxide and nitric oxide, they had a slower rate of decrease compared to the control. This is also true for the stomatoconductance. As the concentration of sodium chloride increases, stomatoconductance decreases with slightly slower rate for the treated plants. And then this is also true for net photosynthesis and evaporation. Interestingly enough, net photosynthesis increased at 25 millimole of sodium chloride, at least for the treated samples. So the control still decreased with a higher concentration of sodium chloride. The third case study is the relationship of gas exchanges in different phenological phases with coffee productivity in the Cerrado in Brazil. Some background information: coffee growing has been expanding in the Brazilian Cerrado. So what they did was a few years ago they had planted 35 different genotypes of Coffea arabica. And then after the first growing season, they organized those plants by growing efficiency, basically by yield, higher efficiency down to low efficiency. And then they use the CI340 to measure air and leaf temperature, PAR, and stomatoconductance, as well as transpiration and vapor pressure deficit. So the results was that variation of the genotypes was observed between the phenological phases. So there was a higher net photosynthesis rate at the different phases from prior to flowering to all the way to fruit fullness. However, despite the differences, they found gas exchange did not explain the divergences in the growth and production responses of the studied genotypes. So yeah, those are a few of the applications that researchers have used our instrument. Again, most of the studies do use stress response as the main indicator for photosynthesis. But here are a few more studies that unfortunately we don't have time to cover, but I thought they were really interesting. Photosynthetic and yield characteristics of winter wheat under 2 rounds of irrigation. This is a 2020 recent study. In fact, all of these studies have been, they were within the 2 years, so I believe 2019. The second is responsive heat tolerant potatoes under different levels of nitrogen. And the last was an evaluation of suitable amount of water and fertilizer for mature grapes and drip irrigation in extremely arid regions. So Susie will be sharing these links along with the other studies that I reviewed in the chat box below. You can also find more studies on our website that utilize the CI340. You can just go to the website and under the applications tab, you can sort the studies by instrument. So in this case, we're interested in the CI340, so you can filter out all the studies by instrument, the CI340, and it'll give you a complete list. The list is always growing. I'm not sure if these are included yet, but we will go ahead and annotate the library to include these as well. And that concludes our webinar for today. In summary, we went over some of the applications and use cases of the instrument. As I mentioned, the CI340 typically is used to investigate the effects caused by stress, but there are also some non-traditional uses as well, such as soil and insect respiration. Who can benefit from the CI340? All researchers in the academic, private, and government sectors that require a fast, reliable, and non-destructive way to measure gas exchange. Many of these studies and research that I went over is in the academic sector, but Stephanie is definitely in the private sector. So those are a couple of examples. We also have USDA has purchased a couple of our instruments as well. Built for accuracy, speed, and reliability, the CI340 provides the means to acquire rapid, non-destructive results with data transparency in mind. And for any support and collaboration opportunities, inquiries, you can contact either myself or our other application scientists, Gail and George. Susie will be sharing our emails in the chat box below. And that concludes for now. We'll go ahead and, well, I actually wanted to talk a little bit about our new CI710 leaf spectrometer. So our new leaf spectrometer has a release date by, Susan can correct me in the chat box below, but I think we have a release date by end of July. The CI710 will have built-in indices as well as room for expansion, so users can add their own indices. But we do have some included from research articles and their for analytes such as chlorophyll, anthocyanins, carotenoids, and more. One major feature that this new CI710 will have is the ability to create PLS models, as well as updating all of your data to the cloud and to our harvest tracking website, Fruit Maps. So this feature is pretty big. It will have some of the same features as the 750, albeit a different spectrometer. And mainly we had in mind thin film applications for this instrument. So we're excited to be releasing that and hoping that some researchers will be conducting some research using our tools. And next, I wanted to talk a little bit about the the F751 Kiwi quality meter. So this is related to the 750 instrument that is widely used in breeding applications as well as commercial use to determine quality metrics such as dry matter, bricks, total titratable acids, titratable acids and total, I meant bricks. But yeah, this instrument will instantly and non-destructively assess kiwi quality and maturity indices like dry matter and bricks. You can stay connected with us and follow us on social media. You can also visit our website to stay updated on new developments, such as the CI710. For sales inquiries, there's a link below. Susie will be linking the link. We'll be sharing the link for to request a quote, as well as requesting any consultations with myself or Galen. You can follow us on Twitter, Facebook, as well as Indeed. We are reamping our Indeed website, so stay tuned and follow us for any updates there. Here's our address, just in case. We encourage any researchers in the area to come and visit. I know in this difficult time that may not be feasible, but I hope everyone's staying safe, and that if you are in the area, once things start clearing up, that we would definitely welcome any visitors. Here again is the link to request a quote, as well as the link to request a consultation. Susie will be sharing these links in the chat box below. And that's the end of my presentation. I'll go ahead and, if you have any questions for me or Stephanie, I'm going to be going through the Q&A box now. So Anthemos is asking, can we measure photosynthesis under LED lights? Definitely. So you can measure photosynthesis under LED lights. I believe Stephanie mentioned that they test and grow under some LEDs, at least some of their plants. So yeah, our PAR sensors have a range of up to 2,400 millimoles. Yeah, I hope that answers your question. Joan Tiramoy is asking, sorry if I butchered your name. Can the CI340 be used for photosynthetic cyanobacteria and algae? If so, are there specific chambers for liquid cultures such organisms? That's a great question. So I believe that you can use the leaf leader chambers. So this would require you to use a closed system. However, so but yeah, I believe that the leaf leader chambers are able to, you would be able to put your bacteria as well as any water into the system. But I would have to double check just because of the moisture level. I'm not sure how that would affect it, but for photosynthetic rate, definitely. I have your email, so I'll go ahead and reach out to you just to make sure. Sanjay is asking, in leaf leader or big chamber, whole small plant, can they be used for ACI curve, temperature curve, light curve, and BPD curve measurement? So in order to have curves, you would have to use one of the optional modules. The leaf leader chamber itself is you can't use it to create curves. But great question. Joe Tiramoy is asking, related to his previous question, also applies to aquatic plants. So again, yeah, you can definitely use the leaf leader chamber, which is just, however, it's restricted to closed system analysis. So, but again, I'll go ahead and ask you or reach out to you to further discuss this application. Sanjay is asking, in the future, CID used measurement of gas exchange and weight bases because it is important for fruit photosynthesis. I'm unsure of the question. In future, CID used measurement of gas exchange and weight bases because your question's a little unclear to me. If you want to go ahead and update that at the bottom to make it more clear. If not, then you can go ahead and maybe we can have a phone discussion about some of your application. I'm not sure exactly what you're asking. Versha is asking, she has 3 questions. Number 1, how can we characterize and measure stress response using the instrument? So I had discussed some research that used stress responses such as salinity and water stress, and they measured the net photosynthesis rate under these stress conditions and compared it to their control. So that would be one way to do so. Her second question, how can we predict the age of the plant using this instrument? That's a great question. I'm not sure that the instrument has that type of predictive capacity. You can definitely, if you know the age of the plant, of course you can measure the photosynthetic rate. I'm not sure if there would be a correlation between age. I'm assuming that there may be, but the instrument itself doesn't make the predictions. Third question, is it useful for soil science studies and carbon sequestration measurements? So using our optional module for soil respiration chamber, you can it would definitely be a useful tool. Thanks for your questions, Versha. Sanjay is asking, in any software to calculate the secondary parameter like RUB-P, TPU, RUB, and a lot of parameters by light curve and VPD curve. Our software does not calculate any of these curves or any of the parameters given by the curves. But great question. Sanjay is asking how to remove CO2 fluctuations in the CI340. So in order to remove some of the CO2 fluctuations, we advise you to be in a stable environment. The ERGA sensor is very sensitive and can measure CO2 concentration from a variety of different sources, including traffic and other sources. What you can do is build a CO2 filter chamber. The instructions to do so are on the support page of the instrument. Susie will go ahead and share the support page to create the chamber. And this will help stabilize some of the CO2 fluctuations that your instrument may be become sensitive to or capturing. Next question, Analytica is asking, can we correlate any plant infection with the CI340? So that would be an interesting thing to investigate. So I'm not sure what infection you would be studying, but definitely the CI340 can determine photosynthesis rate. I would assume that it would definitely have an impact on the photosynthesis, as well as stomatoconductance, especially if it's any infection to foliage. Yeah, that's a great question. I'm sure that there are studies. If you want, I can go ahead and look into that and get back to you with some examples. Go ahead and give me an email so that I can go ahead and send you some examples though. Francisco is asking, on average, how much time does it take to make each measurement? Great question. So the fastest measurement time in differential mode is about 40 seconds. So it takes first 20 seconds in to measure the input CO2, and then the time interval between input and exhaust is set by the user. If you set it to 0, then it would immediately go into the exhaust mode, and this takes another 20 seconds. At the end of the 20 seconds, it takes the differential point and giving you both the input and exhaust differential to take the measurement, or to give you the result. Of course, the user interval can be set by the user, so it really depends on your application, but the fastest it can take a measurement is 40 seconds in differential mode. In continuous mode, it's continuous, so it would just take as long as you want it before you stop the analysis. But great question. Thank you, Francisco. MD is asking, recently I used a CI340 handheld system to measure net photosynthesis rate of tall fescue leaf and growth chamber experiment using open system leaf chamber. However, I found the net photosynthesis were 0, although I saw all other parameters in the CI340 screen. Does it need CO2 connection? Great question. So it may be that the CO2 canister is depleted, but if you want, go ahead and send an email over to support. Susie will go ahead and share the email for the support page. We'll go ahead and need your serial number, and we can go ahead and troubleshoot your instrument to see what exactly is occurring to give you these results. Charles is asking, were readings taken at the same time or any other time so far as the period is up? I'm not sure if he's referring this, or if he's asking Stephanie about this, or I think this was earlier in the presentation. This could probably be a question directed to Stephanie. Again, were readings taken at the same time or any other time so far as the period is up?
So yeah, we take all of our measurements at a standardized time after the lights come on. So again, we're indoors and the lights come on at different periods of time, and we allow the lights to be on enough so that the plant can kind of equilibrate and get to a maximum of, you know, its potential before we go in and take measurements.
Thanks, Stephanie. I hope that answered your question, Charles. If not, then feel free to give me an email so we can clarify your question. MD is asking, do the instruments need calibration before use? Yes, so the instrument comes pre-calibrated. We recommend that the instrument be calibrated every 3 months, but biannually is also acceptable. There are instructions on how to calibrate the instruments in the manual. You can visit our website to access the manual. We also have videos on how to do so. I encourage everybody or anybody to visit our YouTube channel. Actually, Susie will go ahead and share that link. This webinar, as well as past webinars, are located there, as well as webinars and other instructional videos for other products that we have. But I did want to mention, yeah, that this webinar will be shared with everybody after it's completed, and we'll go ahead and send an email to everybody indicating or with the link to the video. But if you are interested, you can definitely visit our YouTube channel to view past webinars as well. Thanks, MD. So Francisco is asking, does the equipment require a startup calibration period, and how long does this take? Great question. So yes, it does require a warm-up period to calibrate the sensors. And this period is about, there's a range of 4 to 30 minutes, which is a awkward range of time. But to be sure, I think 30 minutes is definitely more than enough time to do so. You can also, I forgot to mention that the instrument comes with a tripod mount. So you can mount the instrument on a tripod and just set it in the environment that you would normally be taking your measurements in order to calibrate in that environment. And again, 30 minutes is more than enough time to for a calibration period. Great question. Macoena is asking, when comparing, for example, the effect of 2 treatments in plants, do I have to use the same leaf position? This is a good question. I would say, since this is a non-destructive measurement, you can definitely use the same leaf position. I would refrain from marking the leaf in any way, but you can also use different leaves or different leaf positions as well. I think consistency is probably key in any research, but yeah, definitely, as long as it's in around the same area, I think, yeah, you can use different leaves, but yeah, I think it really just depends on user preference. Great question. Shanil is asking, you mentioned you are looking into nutrients, additives, and that the CI340 is useful to determine stress response. It is also is it also useful to detect stress alleviation by applying additives like plant biostimulants, or are these effects on photosynthesis too small to measure compared to the variation? That's a really good question. I think there have been inquiries by actually researchers to try to study this variation to see if there's any effect on additives. In one of my case studies, actually I did talked about, it was the second one where the researchers had applied the nitric oxide donor as well as the hydrogen peroxide. And they did actually notice an effect on photosynthesis rate. But this was also in under stressful conditions, so that may have played a higher role in it. But I would encourage you to look at that case study again. But as for Stephanie, I don't know if you, I think she had definitely used, I'll go ahead and let Stephanie talk a little bit about this and her experiences.
Yeah, so that is part of our testing. We test products that we hypothesize will alleviate stress for the plant. And we have been able to find statistically significant differences in some of our trials. And some trials we can, some trials we can't. Some of it depends on what the condition of the plants are entering the trial, or if there's any other mitigating factors that happen. But we are able to detect those different stress alleviation differences due to the application of biostimulants.
Thank you, Stephanie. I hope that answers your question, Shanil. Next question, Jose is asking, not to put you on the spot, but my only experience with measuring photosynthesis has been with other well-known companies' product. Not sure if I can mention it, but I will. Lycor, do you have experience with both, and how would you say these compare in data quality? That's a good question. So, me personally, I have no experience using the Lycor photosynthesis meter. Both are great products. Definitely they have a lot of optional modules. However, our instrument is much more portable. I mentioned you can take it it's into the field without lugging additional backpacks or other carrying cases for the modules. But yeah, definitely both provide photosynthesis. Ours, however, is, in my opinion, just much more portable, weighing in at under 3 pounds. Also, all of the data is saved to external memories, which also makes it super portable. You don't have to take the actual instrument into the lab if you don't want. Just taking the actual memory card to your workstation. But great question, Jose. Thank you. Charles is asking, were readings taken at the same time? I'm not sure who this is directed at, but I would recommend that most readings be taken at the same time or around the same time. Just consistency would probably be key if you're doing all of them on the same day or not. But yeah, I think just being consistent would be my best advice. Thanks, Charles. Oh, I think there's additional, or were they taken any time so far as the period or time was up? I mean, I guess that doesn't really change my answer. Try to probably be as consistent as possible. Yeah.
I think they may have been those two questions may have been directed to Stephanie. in
Okay,
regards
yes,
to the work she presented.
Stephanie, did you want to comment on whether the readings were taken at the same time or were they taken at any time so far as the period or time was up?
Yeah, so as I've mentioned before, we try to take them at roughly the same time when the lights in our indoor facility go on, to try to standardize, because photosynthetic rates do fluctuate throughout the day.
Excellent. Thanks, Stephanie. I hope that answered your question, Charles. Anonymous attendee is asking, one of the drawbacks from common gas exchange equipment that I usually face is difficulty to record the data due to fluctuated CO2 concentration. Is there any embedded system software within the CI340 that can tackle the problem? Yeah, so the CI340 does come with soda lime inside of the instrument itself in order to help purge CO2. As I mentioned in a previous question, there is a way to help stabilize CO2 fluctuation, creating a separate chamber that would lead into the inlet of the instrument. On our support page, there's instructions on how to do so. I believe it's the last question in the Q&A or the FAQ of the support page. This will definitely help it will definitely help stabilize the any CO2 fluctuations. Great question, thank you. Amadou is asking, how long does it take to calibrate at the end of a measurement? So the whole calibration process takes about 30 minutes at most. But the instrument doesn't have to be calibrated after each measurement. In order to come back to equilibrium, the soda lime will scrub the CO2 from the system. But if you're referring to calibration that are required quarterly or biannually, the whole process takes about 30 minutes at most. Next question, can we correlate any plant infection with measurements of CI340? I think I answered this question already. Yeah, but if, again, there are definitely some research, there's research that has investigated plant infection, and if there's correlation with net photosynthesis rate as well as stomatoconductance and transpiration. Just off the top of my head, I would probably assume that yes, there's probably some correlation, especially if it's affecting foliage. Sanjay is asking, difficult to control CO2 in the experiments in indoor conditions. I think this is kind of related to some of the other questions on the CO2 fluctuations. There are instructions on how to create a chamber in order to help mitigate these fluctuations. And in an indoor condition, it poses a different type of environment in and itself. It is a little bit more probably stable, but it really depends on the CO2 concentrations of your indoor environment to begin with. Thank you, Sanjay. It looks like Stephanie actually exited the webinar, and so any questions directed to her, she would probably be a lot more capable of answering than I would. So unfortunately, this next question by an anonymous attendee is asking, hi Stephanie, you mentioned that the CI340 is one of the pillars of your robust product testing platform. Are there other parameters or technologies that you regard as essential? I can't speak, unfortunately, I can't speak for Stephanie on this, but she did mention a SPAD meter that she had used. If you have any questions for Stephanie directly, though, Susie will be sharing her email in the chat box below. But yeah, that question, unfortunately, I can't answer. Sanjay is asking, what is the exact period for salinity gas exchange measurement in daytime? This is one-time measurement. For the study that I reviewed, and they had used differential, so it's definitely not continuous. I'm assuming they probably took multiple data points over probably a period of time. I think most research uses about 10 minutes of data in differential mode. But again, I can look into this a little bit more. But if also Susie has shared the links to these studies in the chat below. Susie, if you want to share article number 2 for Sanjay, so that he can maybe there's in the methods section exactly their method for measurement. Great question though. Thank you, Sanjay. Anonymous attendee is asking, what makes the CI340 better than other ergas? Great question. So the CI340, just the portability and field ready use is probably our biggest benefit to our instrument. Other, I can't speak on other instruments, but I know that our portability is unrivaled. And so I think the ergonom itself is a pretty stable sensor. And so there's not much variation between sensors, to be honest. And so I think if you can have something as portable as possible, that probably will give you more flexibility in your measurement and the way that you want to run your experiments. Sanjay asking, high yield coffee direct correlation with gas exchange. In that study, they actually, in this case, they found that there wasn't much correlation actually between it was only 2 harvest periods. So I think once they have more data, there could possibly be differences in their results or conclusions, but I think the result of their study showed that there wasn't much correlation between the gas exchange and a high yield in coffee and the different genotypes of the plants that they had planted in Brazil. But that's a good question. Thanks, Sanjay. Macarena is asking, just to clarify, if I use CI340, I can be able to get all the parameters such as stomatoconductance, chlorophyll fluorescence, photosynthesis rate in one measurement? Yes, definitely. So to clarify, yes, you can get all these parameters. Actually, with chlorophyll fluorescence, you do have to have one of the optional modules in order to do so. However, the other 3 or the other 2 parameters, as well as transpiration, in addition to stomatoconductance and photosynthesis rate, you definitely will get that data in one measurement. Anonymous attendee is asking, can the CI340 be combined with photosynthesis efficacy analyzer based on fluorescence. That's a great question. I'm not sure about that, actually. I would have to look a little bit more into this question in order to answer. If you want to go ahead and give me an email, I can get back to you today about this. Susie will go ahead and share my email in the chat box below. But I appreciate the question. Sanjay is asking, difficulty and stability for humidity. How can I minimize? That's a good question. So I would first recommend that you purge the system of any existing humidity using the silicone beads that come included with the instrument itself. And then in addition to that, then I would probably just monitor the actual humidity. If you have an external humidity sensor, I would use that in coordination with the instrument in order to see exactly what may be the issue with the stability of the instrument itself. Great question, though, Sanjay. You've had a lot of great questions. I appreciate that. Merle is asking, what is the adjustment time for the cuvette conditions for the CI340? Does it have a rapid adjustment technology? I'm not sure exactly what you're asking, Merle. Adjustment time for the cuvette conditions. I would recommend that you probably just send me an email. I'm not exactly sure what your question is asking. Mohammed is asking, we have a CID 70 spectrometer can be correlated with this data with UAV multispectral data indices. Unfortunately, I don't have any experience with the CI70 spectrometer. I think this was before I was in the part of the company, and so I don't have any experience with that. I can go ahead and have a...
I think they may be referring to the 710.
Oh, 710.
Yeah.
Got it. Okay, so you have a 710 spectrometer. Can you correlate the data with UAV multispectral data indices? Currently, you cannot. However, with the new model or new version of this CI710, this may be viable or feasible, but since that isn't released yet, with the current one that you have right now, it won't be feasible. The CI340 is able to detect, measure foliar water uptake or just the outcoming H2O due to transpiration. So that's a great question. It is it's just able to detect the outcoming H2O due to transpiration. Great question, though. Lenovo is asking why we found a negative value for the net photosynthesis. That's a great question. So if you have a negative photosynthesis rate, net photosynthesis rate, that's due to there not being photosynthesis occurring, and it's actually in respiration. That would be one explanation for why. However, if you know that for a fact that photosynthesis would be occurring, then it could be a support question that you may have to contact us with, and Susie will go ahead and share that email in the chat box below. Go ahead and send an email over to support with your serial number, and we can go ahead and discuss. But definitely, if it's not uncommon that a negative value would exist, and that's due to respiration of the plant. Mohammed, how many plant samples should be taken for measurement of spatial distribution? I don't know if that's really. I mean, I could have my recommendations, but I wouldn't really. I'm not in a position to advise, I guess, how many samples. I think there's probably a way to figure out at what point any more samples don't actually contribute any more statistical significance. But off the top of my head, I don't have that answer. But thanks for asking, Mohammed. Sanjay, in fluorescence, can we measure directly QP, QN, NPQ, ETR, and CI340? That's a great question. I think actually I would have to double check with some of these. I'm not sure if all of these parameters are measurable, but go ahead and send me an email, Sanjay, and I can double check that for you. Richard is asking, the leaf spec sounds really interesting. What spectral resolution and or number of bands does it have? I'm glad that you're interested, Richard. Yeah, I'm super excited about this instrument as well. The spectral resolution is this 1 nanometer, and the range that it would have is 300 nanometers all the way up to 1200. And with, yeah, of course, with the spectral resolution of 1 nanometer. We don't have, I'm not sure if we have a brochure with more information on the unit itself. You can, I believe it has similar specifications to our previous unit, which you can find on our website at cid-inc.com. Susie will go ahead and share that link with you in the chat box below. And if we do have a brochure, I'll go ahead and send me an email, and I'll go ahead and double check that for you. I don't know if Susie
brochure isn't finalized yet,
okay.
But it should be in the next 30 days.
Okay, yeah. So
and we will be hosting a webinar when that instrument is officially released.
Definitely. So thank you, Susie. Talk to him is asking, can we use CI340 to measure photosynthesis of plants in tissue culture? Definitely. So you can definitely use it in tissue culture. As again, one of the main benefits is non-destructive form of measurements. You can put the entire tissue culture within a chamber in order to do so. Great question. MD is asking for calibration. Do I need to send the instrument to you, or can you perform it yourself? So the calibration, the user is able to do the calibration themselves. All it requires is a standard gas in order to do so. And there are instructions on how to perform the calibration on our website as well. I would think there is a video as well on our YouTube channel as well. But that's a great question. Thank you, MD. So Rob is asking, how can I take measurements from maize? So you, it really depends. Are you asking, you want to take measurement of the maize itself, the seeds, or are you or of the husk. You could definitely, I think one of the studies that I shared deals with maize actually, and so you can go ahead and look into their research article to see their methods for measuring their maize plants. But I think they were measuring the husk itself. Susie, I think, probably shared the links to the 3 other articles that I didn't have time to cover, but if not, she'll go ahead and share those now as well. Olga is asking, how often do you have to change the CO2 canister? That's a great question. So it is about, I would recommend every year, but it's known to actually last longer than that. Up to 3 years is, in my experience, the longest the CO2 would last, but this also really depends on use of the instrument. But there are links to ways to purchase CO2 canisters. They're common canisters that you can actually acquire from most suppliers, and if Susie can share the link to that, it would be from our support page. If not, you can visit our support page under FAQs. There's a section that talks about the CO2 canister, and in that section, there's actually links to suppliers of our CO2 canister and the specification for the actual canister. Thank you, Olga. Swarov is asking, how can I measure photosynthesis in artificial light? Like LED lights, you can measure photosynthesis just as the normal way that you would typically measure photosynthesis. Stephanie had mentioned that at her facility at Arcscreen Technologies, some of their plants are actually grown under LEDs. And so that hasn't been an issue for them. As well as you can also use one of the optional modules, the light control module, in order to control the light environment that you will be taking the measurement in. Great question, Swarov. Krishna is asking, are there any studies where CI340 is used in ornamental woody plants? Off the top of my head, I do not have the answer, but I can almost guarantee that there are. Just based on the request for pine needle chambers. And so the pine needle chambers are commonly used in conifers. And so I would assume that, yes, there are probably studies. You can visit all of our studies and library of studies on our website. On the application tab, you can search or filter out studies by instrument. Just go ahead and click CI340, and it'll bring up our entire list of studies, as well as additional studies that we're constantly adding to that. Anonymous attendee is asking, have you compared data between your system and Lycor portable photosynthesis system? No, we don't really compare ourselves to competitors in that way. I think that's probably unfair to both systems. They are photosynthesis is pretty standard, and so it's both will offer results. We pride ourselves on the portability and ease of use of our system, as well as the data transparency that comes along with that. All of that, our data comes, all of the raw metrics as well as the calculated results are available for users to use, and I think that's kind of where we stand on that. So Rob is asking, can I take whole canopy measurements in lentil crop? Yes, you can. Anonymous, have you tried the CI340 in real field conditions? How do you handle fluctuations in PAR, temperature? Do you think it would be reliable to compare stress responses in a trial with higher number of plots? Researchers have definitely used the 340 in real field conditions, not just controlled indoor conditions, but in real field conditions. All of the studies that, especially the third one in Brazil with the coffee beans, that's a real world application. And everyone has their own way to deal with fluctuations in PAR. Of course, you can't control the weather, but and light and temperature. So I think the best way would probably be just to be as consistent as possible. Try to take measurements at the same time of day in order to mitigate some of that variance. That's probably what most people do. Great question though. For chlorophyll fluorescence, have you tried the CI340 in real field conditions? Just answered that. I think that's a duplicate. Last question, anonymous attendee. Have you tried two CI340 devices in the same experiment? Have you obtained a good correlation between them? That's a good question. I personally haven't, and I'm unaware that if anyone else has, but the erga sensor is very stable, and so you can expect very little variation between that. If anything, there may be just, it really depends on, you can't be taking measurements exactly in the same position with two instruments. The closest you could do would probably be side by side. And so it's probably acceptable that you would see slight variance between instruments. But to answer your question again, I personally have never used two at the same time, and I'm unaware of any researchers that have either. Okay, this is going to be the last question. After clamped a leaf, how long does it take to get a stable measurement? Within a minute? Yeah, so the quickest measurement you can take is 40 seconds. You can set an interval, a user interval between intake and exhaust of CO2. And so you can set that in order to, I don't know, possibly stabilize a little bit more, but yeah, I think each measurement or data point takes 20 seconds with the entire net photosynthesis result being obtained in 40 seconds at minimum. So that concludes all the questions. Those were really great questions. I appreciate everybody's time for attending. I know these are difficult times due to COVID-19, but I hope everybody is in a safe location, practicing safe habits. And I really appreciate, again, everybody in attendance. Those were great questions. All links are shared in the chat box, as well as Stephanie's email. If you have any questions, regarding her cannabis research. It's an exciting industry to be in, and I appreciate you attending again.